This lecture will cover the process of protein synthesis. In the nucleus are chromosomes. Chromosomes contain DNA. DNA stores the genetic instructions for proteins. Proteins determine cell structure and function. DNA is the instructions for every protein in the body. DNA is organized into genes. In general, one gene codes for one protein. So one gene is a segment of DNA carrying the instructions for one polypeptide chain. These instructions are carried in the genetic code. The genetic code is the chemical language of DNA. This is the sequences of A, T, G, and C. Remember, A stands for adenine, T stands for thymine, G stands for guanine, and C stands for cytosine. These are the letters of our genetic code. These letters are arranged into triplets. 3 bases, which is each base is one letter. 3 bases are going to code for one amino acid. So these are going to be the words that specify a single amino acid. The sequence of triplets in each gene forms a sentence that describes how to make a protein. So each sequence of triplets is going to describe the number, the kind, and the order of amino acids. The process of protein synthesis not only includes DNA, but it also includes RNA. Remember, there are 3 forms of RNA that work to carry out DNA's instruction for protein synthesis. First, there is messenger RNA, which is abbreviated mRNA. Messenger RNA is a single-stranded copy of DNA that carries the DNA message or instructions out of the nucleus to ribosomes in the cytoplasm. Messenger RNA is used both during transcription and translation. Messenger RNA is required because DNA cannot leave the nucleus. So messenger RNA copies the gene and carries that copy out of the nucleus to the ribosomes in the cytoplasm. Second, there is transfer RNA. Transfer RNA is abbreviated tRNA. Transfer RNA is a small cloverleaf-shaped strand of RNA. One end of transfer RNA binds to a specific amino acid. The other end of this tRNA is going to match to a codon of mRNA in the ribosome. This will be explained in more detail later. Transfer RNA is used during translation only. Finally, there is ribosomal RNA, or rRNA. Ribosomal RNA is found in a ribosome, and it helps to translate the messenger RNA into a protein strand. Ribosomal RNA is only used during translation. In order to understand the process, we are going to need to discuss some vocabulary, beginning with codons. Codons are a 3 base pair sequence of messenger RNA that matches the DNA triplet code. So the 3 base pair sequence in DNA is called a triplet code. The same 3 base pair of sequence on mRNA is called a codon. A codon is going to code for an amino acid to make the protein strand. There are 64 codons total. Each one codon codes for only one amino acid. Now, you may remember that there are 20 amino acids, which means that there is some redundancy. Now, I told you that one codon codes for only one amino acid, but because there are 64 codons, 64 codons can't code for 20 amino acids and have no redundancy. So each amino acid may be coded for by multiple codons. In your PowerPoint, I provide a codon chart so we can see our 64 codons and the 20 amino acids that they code for. So for example, UUA codes for the amino acid LEU, which is leucine. However, leucine can also be coded for by UUG, CUU, CUC, CUA, and CUG. So there are 6 codons total that code for leucine. However, each of those individually will only code for leucine. So every time there's a UUA, a leucine will be put into place in the sequence of amino acids. This redundancy acts as a safety net against mistakes and mutations. So if in your mRNA strand there should be a CUU and instead there's a CUC because there's a mistake or a mutation, you are still going to put LEU in the correct space in our sequence of amino acids. This guarantees that our sequence of amino acids will stay the same despite any mistakes. Remember, if you change the sequence of amino acids, you change the function of the protein. So redundancy acts as a safety net. There are several different special codons that you need to be aware of. However, you only have to remember one of those specifically. The one that you need to remember is the codon AUG. AUG is called the start codon for all proteins. That means all proteins initially start with the codon AUG. If there is no start codon, the protein will not be made. Similarly, all codons need to end somewhere. There are 3 stop codons that tell the ribosome when to stop attaching amino acids. These 3 stop codons are UAA, UAG, and UGA. You do not need to remember those specifically. However, whenever a ribosome reads a UAA, it is going to stop attaching amino acids and that protein is finished being made and the ribosome will release the finished product. I'm now going to give you 2 different versions of protein synthesis, a short version and a long version. Each of them contains 3 major steps. The first step of our short version is gene activation. Gene activation means that the DNA is going to uncoil, the DNA strands separate, and RNA polymerase is allowed to bind to the DNA strands and start building the mRNA. RNA polymerase sounds like a big, fancy, scary word, but it's really not. Let's break it down. RNA polymerase. Let's start at the end. ACE tells us it's an enzyme. A polymer is a strand of something. Well, it's RNA polymerase. So RNA polymerase is going to make a strand of RNA, more specifically of messenger RNA. This is the process of transcription. Transcription is step number 2 in our short version. Transcription is going to copy instructions from DNA into mRNA inside of the nucleus. Transcription uses the exact same language of nucleotide bases. We're going to go from nucleotide bases of DNA into nucleotide bases of mRNA. And that's it for transcription. Our third step is translation. In translation, ribosomes are going to read the codons of mRNA in the cytoplasm and make proteins. Translation uses different languages. We're going from the language of nucleotide bases in mRNA to amino acids of our proteins. That's the short version of protein synthesis. The long version is not much more complicated. What happens is gene activation is going to occur. So here's step one. Step one is gene activation, where our DNA again is going to uncoil, and our RNA polymerase is going to copy DNA into mRNA. Now what you need to remember is that RNA uses slightly different base pairs than DNA. DNA uses A, T, G, and C, adenine, thymine, guanine, and cytosine. RNA uses A, U, G, and C, adenine, uracil, cytosine, and guanine. So in RNA, we have traded the T for a U. When RNA polymerase is making mRNA strands, each time it comes across a T in the DNA, it will make an A in the mRNA. Each DNA C becomes an RNA G. Each DNA A becomes an RNA U, and each DNA G becomes an RNA C. So during this process, C matches to G, G matches to C. T in DNA is going to make an A in RNA, and an A in DNA is going to make a U in RNA. Once the RNA polymerase is finished copying DNA into mRNA, that mRNA needs to go through RNA processing. This is where the RNA is edited and unnecessary portions called introns are removed from the mRNA strand. The good portions, called exons, are going to exit the nucleus to go to ribosomes and make a protein. So introns stay inside of the nucleus, and exons exit the nucleus with the mRNA. When the mRNA exits the nucleus, transcription is complete. As the mRNA encounters an rRNA in our ribosome, translation begins. Remember, translation is using two different languages, and we're going to go from the language of nucleotide bases to the language of amino acids. This entire process is going to occur in the cytoplasm and involve all three types of RNA. So mRNA moves into the cytoplasm and binds to two ribosomal subunits. The transfer RNA, or tRNA, is going to deliver the correct amino acids to the ribosome and match that amino acid to the codon of our messenger RNA. Enzymes are going to join amino acids in order with peptide bonds. So our ribosome is going to start reading our mRNA strand, and when it finds the codon AUG, the ribosome begins to attach amino acids. Each codon is going to be read by specific tRNA binding sites in our ribosome. These tRNA binding sites are going to provide areas for the tRNA to match its amino acid to the codon of the mRNA. The ribosome then moves down the mRNA strand, attaching the correct amino acids delivered by tRNA. When the ribosome reads a stop codon, it will not attach any more amino acids. The ribosome comes apart into its two ribosomal subunits, and the completed protein is released into the cytoplasm. At this point, translation is complete. So as a review, protein synthesis is going to take the genetic code of DNA, and in transcription, DNA triplets are transcribed into mRNA codons by RNA polymerase. In translation, these codons are going to match tRNA, and tRNA will deliver the correct amino acids to the ribosomes. Amino acids are peptide bonded at the ribosomes to form polypeptide chains. Start codons and stop codons are used to start the process of translation and in the process of translation to complete making a protein. Now, it's very easy to confuse transcription and translation, so what I recommend that you do is remember that they're alphabetical. They both begin with trans, T-R-A-N-S. However, the very next letter is different. C comes before L in the alphabet, so transcription occurs before translation in the process of protein synthesis.